Hi, it's Mrs. Clark. Let's review protein synthesis. So protein synthesis starts with DNA. Remember, DNA is the master set of instructions for everything that happens in your body. And it performs those functions by making proteins. Now, DNA never leaves the nucleus, in eukaryotes at least, because prokaryotes do not have a nucleus. And so the question becomes, how do we get this information needed to make the proteins outside the nucleus? And that's where step one of protein synthesis comes in. Step one is called transcription. And it involves a RNA molecule called mRNA. M stands for messenger, because this mRNA is going to get the information from the DNA strand, and then it's going to take it outside the nucleus. And so transcription is when we are actually copying this information from DNA into our mRNA. And then our mRNA strand is going to leave the nucleus and go somewhere called the ribosome. And this is where step two of protein synthesis happens. Step two is called translation. And this step involves, of course, our mRNA, which is now going to be our instructions because it copied the instructions from the DNA, remember? And another RNA molecule called the tRNA. Now, the tRNA's main job is to bring in the amino acid. Remember, amino acids are the building blocks or the monomers of proteins. And so the tRNA is basically bringing in the supplies needed to make the proteins. And so you may be wondering, how does the tRNA know which protein goes next? Remember, our mRNA strand is composed of nitrogen bases, or a sequence of letters. And these letters are read in threes. We call these threes codons. Now, on the tRNA molecule, there are also three nitrogen bases, and this is called the anticodon. And the anticodon is going to match up with the codon, and that's how it knows which amino acid goes next in the sequence. And so eventually, it's going to build a sequence of amino acids, which we know to be a protein, which is our final goal in protein synthesis. So this is protein synthesis. We refer to this whole process as the central dogma. So both transcription and translation are called the central dogma together. Now, when you're performing these problems, be very careful when you're transcribing your DNA into mRNA. Take your time, because any errors made here will carry through the rest of the problem. Okay, now when you're performing translation, be very careful to not use the anticodon on your codon chart. It's called a codon chart. You must use the mRNA strand. If you use the anticodon on the codon chart, your answer will be wrong. Now, a good hint for remembering which of these comes first is that C comes before L in the alphabet. So if you can remember C comes before L in the alphabet, maybe you'll have a better shot at remembering transcription comes before translation in the central dogma or protein synthesis.